So as we did in the last class, first I'll be revising some basic topics, and also just to inform everybody, somebody in the last class had mentioned that they want to get a little bit of a recap on cell signaling in general. So that is what I've kept the focus on for today. We'll revise the basics because if that is not clear to you, it will be very difficult to catch up eventually. So we are going to clear basics today about cell signaling, about the different types, the different components, and then I'll complete whatever was left over from the previous class about the TP53 signaling pathway, and a little bit about the druggability of it and what was initially scheduled to be discussed today. We'll see if we can cover it today. Maybe we'll just have a discussion about the steroid hormone receptors and examples of the same. If we run short of time for it today, then we'll strip that for the next class. And along with it, we'll move on to diagnostic techniques because otherwise we'll run short of time in the long run. But don't worry, if anything is left behind, we can schedule one extra class of maybe an hour or so, and towards the end, and we'll have a discussion on whatever is left behind. And you can bring up any queries. Don't worry that if there's only like 12 classes that are scheduled, there can only be so much discussion. No, we can have one extra class in the end, and we can clear out anything that is left behind during the entire tenure of the course. So, what I wanted to start off with, first of all, was a quick recap as we usually do for the course. So just to remind everybody, please let me know if you can't see my screen at any point of time. Just to remind everybody, this was the content of the overall content of the course. So we have discussed hallmarks of cancers and how cells become cancerous. We are currently discussing the second topic, key signaling pathways altered in cancer. We discussed some basics, not basics, even some types of signaling pathways in the previous class. We will go a little backward, discuss basics today, and then cover just one more pathway for today. And if possible, I'll have a discussion about some other pathways as well. Then we will have to move on towards detection of cancer. First, we'll have a discussion about traditional methods. We'll try to cover this in the next class, and then we'll move on to advanced techniques, and then combinatorial therapy, the idea of precision medicine, cancer statistics, etc. All of this. So don't worry, it looks like a lot. It'll get covered in time. And so that is the basic structure. So yes, again, to remind everybody, if there are any doubts at any point of time, you can unmute and ask me, and I'll try to clarify them to the best of my abilities. So basically, last week, what we had done is gone into some details about some topics. We had discussed some key components of the signaling cascade. I will be showing more simplified versions of this today, so don't worry. Then we had, sorry, yeah. Then we had looked at the concept of different feedback loops that operate in cancer, starting off with feed-forward loops, which are necessary, especially present in lower organisms. When I say lower organisms, less complex organisms is what I mean, like E. coli, for example, whereby to suddenly increase the expression or the amount of a certain protein within the cell, a multi-pronged approach is taken. For example, where the default pathway is to trigger the activation of B by A, and then this chain goes on forward. There is also an additional pathway that operates to cumulatively help the process go a lot faster, and for the levels of that protein Z in this example to increase very abruptly. And that is what is relevant for the condition that the cell is in. Then we talked about positive feedback loops and negative feedback loops. We talked about what goes wrong in cancer. We talked about oncogenic signaling pathways. Okay, I have a different version of the PPT. Just give me a second. So I had edited this PPT later on. So I'll show the one that we had discussed in class. Yeah. This is the one we had discussed. Yeah. So we had talked about all these different sort of loops that are present: feed forward loop, positive feedback loop, negative feedback loops, and the definitions of the same, some differences between these pathways and the type of regulation that is maintained, and how these pathways, um, apart from these pathways, an overall loss of control that goes wrong in case of cancer. So we had discussed all of this. Then we moved on to an example of how scientists identify these altered signaling pathways. And I gave you this example of this particular paper published in the journal Cell in 2018, where they had taken patient samples, where they had taken the information retrieved from patient samples, which is stored in a repository known as TCGA, or the Cancer Genome Atlas. And they had analyzed tumor samples, which are of 33 different cancer types. Some examples and some abbreviations were shown here. And they had looked at what are the different types of alterations that occur, what are the key pathways that are found to be altered. And in this paper, they have also discussed some cancer drugs as well. But since we haven't gone into that yet, I did not really bring it up today, uh, even that day for that matter. So basically, what I wanted you to understand here was that how do you actually figure out how cells are, facing changes or are dealing with the changes that occur in these signaling pathways in cancer. Then we had gone into details about the first pathway, which was the RTK-RAS signaling pathway. And we had seen how there are several sub-steps which occur, starting off with the fact that you have a signal molecule which binds to the receptor. Here, the signaling molecule is the ligand, which is extracellular in nature, which binds to its receptor. Upon receptor binding, there's receptor dimerization, as a result of which tyrosine kinase is activated, autophosphorylated, and then the signal is relayed to the downstream proteins. This is the basic change that happens right at the top of the cell, near the cell membrane. Then we looked at how it plays an oncogenic role. So to understand that a little further, we had seen the different types of changes which occur. Now, how do you understand those changes? You understand that by looking at what happens inside the cell after the receptor activation has occurred. So under that, we had seen that a lot of downstream changes occur, starting off with phosphorylation of several other proteins, including the MAP kinase pathway proteins like RAS, RAF, MEK, ERK, all of these downstream proteins. So these changes occur, and apart from that, a separate pathway, which we have also discussed slightly in the upcoming slides, which I'll be showing as well, whereby we saw the activity of phospholipases and how PIP3 helps in activation of AKT, and that leads to activation of mTOR, and that has an effect over the overall cell survival. Not just survival, it also affects other pathways like proliferation, differentiation, cell cycle control, cell migration, etc. Then we saw what exactly goes wrong. So there are lots of ways in which things can go wrong. This was just a reiteration of the same. I'll move past it. So the oncogenic effect can be manifested by a number of different possibilities, starting off with the fact that there can be overexpression, which is an increase in the expression of the receptor at the surface. That overexpression can occur in one of two ways. Either it can be because of increased transcription and obviously translation of that gene, or it can be because of gene amplification, which means that the same gene has undergone duplication multiple times, as a result of which they're all present one after the other. So instead of there being one copy, there are, say, 30 copies of that same gene. I want you to go and trace back, if you have not already studied it, the example of a neurological or rather a neurodegenerative disorder that happens because of these sort of duplications. Keep in mind this duplication, the one that I'm talking about, that is not actually it's not a gene duplication. It is the duplication of a small unit. I think I'll even mention it. CAG repeats. So C, A, and G are repeated one after the other. And this duplication obviously occurs in the DNA. See what happens as a result of this duplication and figure out the name of the disease and let me know in the next class. If you're already aware of it, you can unmute and discuss it with me now. But yeah, it's an interesting disease. It's not cancer, obviously. Otherwise, I would have discussed it right now. But yeah, it's another example of how duplication or tandem presence of a particular um, region can lead to diseases. Apart from that, we also saw that activating mutations within the receptor can also lead to can also be oncogenic in nature. Then we learned about oncogenic fusions, whereby different chromosomes or separate parts of different chromosomes, they combine or they fuse with each other, as a result of which the new protein that is formed as a result of that fusion is an abnormal protein. Now, this abnormal protein can either be completely removed from the system, it cannot have any effect, or it can be oncogenic in nature. That oncogenic nature can either be such that this receptor now has become modified in such a way that it is still present on the surface, but the cytosolic part or the intracellular part of it is different. That difference either makes it completely inactive, that's also a possibility, right? Say, for example, here, if the ligand binds, even if the ligand is binding, nothing is happening downstream of it. So it has shut off a pathway. Now, the shutting off of a pathway can sometimes also be very dangerous. Why so? If it is a pathway that is necessary to maintain a negative feedback loop within the cell, then if there's a loss of activation of that pathway, then it can lead to cancer as well. The other possibility, which is something you see a lot more commonly, is that the ligand, the receptor has become modified in such a way that it is no longer dependent on the ligand. As a result of which, this modified protein is always active, and this modified protein can keep on switching that pathway on irrespective of what is happening inside the cell. So this was another example of an oncogenic fusion. Finally, we also looked at ligand dysregulation, where different types of ligands are able to bind to the same receptor and they're able to elicit activity. Sometimes this is also called as ligand, receptor promiscuity, promiscuous behavior, which means non-specific behavior. So instead of the receptor being very specific for one class of ligands, what it instead does, or rather one type of ligand, it is able to bind to similar types of proteins. Say, for example, I'll give you a very common example, which again is from prostate cancer. So the receptor AR, androgen receptor, is often able to bind, instead of just binding to its cognate receptor. Again, these are jargon. Cognate means, cognate is the terminology which is used in biology, in this sense, in cell biology and molecular biology, whereby it means that if there is receptor X, then it should bind to ligand X only. So there's a specificity between the two. So instead of binding to ligand X, if the receptor is able to bind to, say, X dash, which means a similar but yet not the same ligand, then that behavior is known to be promiscuous. So the example I was giving, so instead of binding to one type of hormone or one type of androgen, it is able to bind to other types of androgens as well. So that is ligand dysregulation that I was talking about. So these are some possible events that can occur. Keep in mind, it's not an all-inclusive list, and a lot of other things can also go wrong, and they can also be equally disease-causing. Uh, then we saw this in a little more detail. We saw that there can be gain of function mutations, amplification, like I mentioned, chromosomal rearrangement, like we saw, duplication of only the tyrosine kinase domain so that it becomes independent of any extracellular stimulation. And finally, also autocrine and paracrine activation. If you remember this example, it was because of whereby multiple cells which are close to each other, one cell is able to activate another cell, and that cell is also able to activate itself. The ability of a cell to secrete a ligand which will bind to its own receptor is known as autocrine behavior, or the signaling pathway is known as autocrine signaling. When a cell secretes a ligand which binds to the receptor of a nearby cell, then that is paracrine binding or paracrine signaling. Is it clear? If not, please interrupt me. Please let me know. I might be going a little fast, but because we have discussed this, so I'm just brushing through the topics a little superficially. But if not clear, please interrupt me and let me know. Then we had started discussing about the PI3K pathway as well. And we saw that this is also an RTK-RAS dependent pathway, but it is not going via the route we had just discussed. Instead, what happens is that after the ligand has bound to the receptor tyrosine kinase at the cell surface, then there's the downstream activation of PI3 kinase. As you understand, the name kinase means it will phosphorylate something. So it phosphorylates PIP2 into PIP3. So this PIP3 then is able to bind or is able to scaffold or allow, like it creates a location or a surface where AKT can bind. And then phosphorylation of AKT is able to do a lot of things downstream of it, which includes survival, growth, proliferation, etc. So this is the key pathway. And the regulation of this pathway is maintained by P10, which is a phosphatase. So these terminologies must be very clear to you. Kinase is something which adds a phosphate. Phosphatase is something which removes a phosphate. Keep in mind that the suffix of ase always means it's an enzyme. It must do some sort of an enzymatic activity. So kinase was doing the addition of phosphate. This is doing the removal of the phosphate. What does that do? It again restores the homeostasis of the cell, or it restores the balance between PIP3 and PIP2. And therefore, PIP2 is again available for another round of phosphorylation to take place so that the signaling pathway can be controlled in a loop when required. When required, why am I saying that? That is because if you just trace back the what we have discussed, then the pathway depends upon the binding of insulin or the binding of whatever growth factor is necessary to the RTK to trigger this downstream pathway. So the final control is coming from outside. The final control is coming from the ligand that binds to it. So that is what we saw, and very superficially we saw this thing about how AKT has different downstream targets, all of which are responsible for different types of pathways. I had highlighted a little bit about MYC, discussed how it is an oncogene, and now it has a huge range of horrible effects that it has on the cell. And when I say horrible, from a normal cell's perspective, but from a cancerous cell perspective, MYC is a wonderful oncogene to have because it has A, it's an oncogene, of course. It causes cancer. B, it is very commonly found to be amplified in cells which are drug resistant. So after you treat cancer cells with say a drug X over a period of time, the cells, by virtue of mutations, changes, etc., over time they develop resistance. So that when we say this development of resistance, it's because it happens due to some changes, some molecular changes, which are occurring within the cell. Now, these changes include one of the main changes in most cases, in most types of cancers and most types of drug resistance is found to be amplification of the MYC gene, which means that more number of MYC copies are synthesized within the cell, and MYC-mediated signaling is restored, which allows the development of drug resistance to emerge over time. So I think we ended at this point. Yeah. So I'll just go back to the PPT that I was showing you guys, and then we can have the discussion for today's class. Right. This is what we'll start off with. Anybody has any questions? Anything you want to discuss, just let me know. One of you just unmute and let me know, and then we'll get started for today's class.
No, ma'am, I don't have any doubt.
Okay. If anybody else does, you can just interrupt me at some future point. Okay, so just to reiterate, we will be discussing basic concepts today, A, because we are a little short of time, and B, because it was required, and I want you to understand this better. If you haven't studied cell signaling before, then I very, very highly recommend you to look at this book, to study, not just look at, to study from this book, Principles of Biochemistry by Albert Leninger. It is the God book for learning any topic. At least initially. You can always have more and more knowledge from multiple books. But any topic, any basic molecular biology, not molecular biology, biochemistry topic that you want to understand, it's a wonderful book too. Cell signaling, most of the things that I've shown here today will be actually taken from Albert Leninger, and a lot of things have been taken from slides that I was taught from in my college by my professor, and they've been credited for the same. But even then, I recommend you to strongly strongly learn from books and from papers once you have understood how to study papers. But yeah, definitely look at this book. Okay, so coming back, we are going to go to the very basics, but we'll understand it once and have it cleared. So, what is cell signaling? The conversion of information into a chemical change, which is known as signal transduction, is a universal property of living cells. So, organisms they use these evolutionarily conserved mechanisms. Why? Because they want to detect what is the extracellular signal, and their job, which is mediated by this process of cell signaling, is to transduce them into intracellular changes. So you take the information from outside, and what you do is you make it, you respond to it on the inside via some intracellular changes. Now, when we talk about extracellular signals, they can be of lots of different types, starting off with antigens. What are antigens? If you've studied immunology, you would be familiar with it. But basically, antigen is part of a, okay, it is a majorly under most circumstances, it is something which can bind to an antibody. The simplest definition of an antigen is something that binds to an antibody. However, it may or may not be able to elicit an immune reaction. What is an immune reaction? That means that when an antigen enters your body or any living system's body, any immunocompetent living cell's body, then what it will be able to do is elicit a reaction to it. That means the entire immune system will fire up and will recognize that as something foreign. So that is the basic definition of an antigen. I'm not going into terminology details because that's all part of immunology, and I'm sure there are courses on it. If you want to learn a little bit about it, you can go to I have some videos uploaded of it from another set of courses that I used to teach before. So you can refer to it. Otherwise, this just Google it and you'll understand the details. Apart from that, other signals to which cells respond include cell surface glycoproteins, oligosaccharides, developmental signals, ECM components, growth factors, hormones. This is what we have learned, light, mechanical touch, etc., etc., etc. These are all other types of signals as well. Now, basic idea of any cell signaling involves these four properties. What are those? First, specificity. Again, you might think that I just told you something that is completely the opposite of it. A few minutes back, I told you about promiscuity. But don't worry, this is the most conventional form that when you talk about cell signaling for a normal cell, this is what is expected, that there should be a specificity between the receptor and the ligand that is binding to the receptor. So the signal molecule should ideally fit its binding site on the complementary receptor, and other signals should not fit. However, there is of course a lot of it's not as final as the way it is written, but in fact, there is a lot of promiscuity that happens, and it is often necessary for the cell to actually survive under healthy conditions, even under in its physiological state. Second is signal amplification. Now, this is very important. You might think it might cross your minds that why is there a need for so many sub-steps to be present in each pathway? For example, when we talked about, say, for example, EGF, we talked extensively about EGF in the last class, which is epidermal growth factor. Why can't there just be EGF binding to EGFR, its receptor, and then downstream of it, it is able to directly allow some transcription or translation to occur? That is because one molecule of EGF binding to one molecule of EGFR, the receptor on the surface, would only, if this was the setup that was in place, if you were to be the designer of nature and you were to design a cell, and if you had this installed, then that signal is not enough to actually help a cell to to respond to that signal that has come in. So only one molecule binding on one ligand and receptor interaction is a very weak signal. So what would be the problem? Why can't we respond to a weak signal? That is because a lot of times, if the specificity of the system is lost, then that weak signal might just be an error. It might be a chance event that a particular ligand has bound to the receptor. And responding to that chance event can potentially be very expensive for the cell. Again, if you remember when I said expense, it means that energetically it might be a very expensive procedure for the cell to actually start off with. So therefore, what nature has put in place is that there must be amplification of the signal, which means that even if one molecule of EGF is binding to its receptor, to To ensure that that binding is correct and accurate, there are multiple sub-steps that have been put into place. Only if that binding is correct will EGFR be able to allow the next step, which is RAS and TRAC, to come in and then make or all of those pathways to be activated downstream of it. And what it does is it increases the weightage of the signal that has come in, and therefore the amplified signal is able to trigger the response a lot faster. So when enzymes activate enzymes, the number of affected molecules in the entire cascade increases geometrically. Here, geometrically basically means GP, geometric progression, not shape-wise, in an enzyme cascade. So signal is binding to enzyme 1. Enzyme 1 is able to trigger enzyme 2, and multiple enzyme 2s are activating enzyme 3, and so on and so forth. The effective net result is a lot stronger. Third property or third characteristic feature of any signaling pathway is the fact that there should be desensitization. Nobody thinks about it. We just talk about the fact that a ligand should bind and the pathway should be activated. No, there should be non-response, or there should be a lack of response to a particular ligand after a definite point of time as well. So desensitization or adaptation tells us that receptor activation should trigger a feedback circuit that shuts off the receptor or removes it from the cell surface. Therefore, the response is under the control of the signaling pathway itself. And therefore, when we say control, it means that it is able to keep a check on how long that signal should persist or how long that pathway should be activated, and shuts it off at the right time. And fourth is integration. This you might not have come across, at least not in my lectures, but basically what it means is that there are multiple signals which are converging down into the same pathway eventually. That is integration. Here, what they're saying, when two signals, they have opposite effects on a particular metabolic characteristic, such as the concentration of a second messenger X or the membrane potential. The regulatory outcome results from the integrated input of both receptors. Keep in mind, this is not something that happens all the time. It's just an example of a signaling of a property of signaling pathways that can often happen. So signal 1 and signal 2, the net result of it is something different. Either it can also sometimes be a more enhanced response, which means that if signal 1 is leading to a response which you can quantify as 2, and signal 2 is leading to a response which you can quantify as 3, then 2 plus 3 might be 5 in some cases, which means it's a much more enhanced signal or much more enhanced response. Or it can be the opposite as well, that 2 and 3 together are shutting each other out. And as a result of it, only the net result is 3 minus 2, 1, which is the response for the second signal, and the first signal is getting shut off. So this I'm just, don't take these exact numbers as a statistics or something to be taken very very particularly. But here's just an example of the fact that signal integration does occur. And apart from that, what has not been shown here is that for any signaling pathway, crosstalk is very, very crucial. That means that multiple signals, even though it was not shown in the example, between multiple signals, crosstalk does occur, and it is very necessary and very extensively present within the cell so as to facilitate faster responses to the ligands that come and bind to the cell surface. Okay, now we are looking at, we are moving forward from the basic definitions and looking at the types of cell receptors. I had mentioned it very briefly in the last class, but yeah, this is again a very composite slide, so I'm coming to it. This is most probably taken from Leninger itself, but yeah, it was part of my professor's slides, so I've printed for the same. So basically, the types of cell receptors can be grouped into all of these different subtypes. The first one talks about a gated ion channel. Keep in mind the terminology receptor, terminology channel, they are all slightly different. They're very closely, often very closely used, like very interchangeably used. But yeah, there are subtle differences between them. So a receptor, as the name suggests, is something which is able to receive a signal. A channel also binds to the signal that has bound to it, but it is the name channel indicates something else. If you look very carefully at this diagram here, you will see that there's an ion which has been shown to go right through the channel. That is what a channel defines. So think of it um, like a pipe, like similar to a pipe. It's not the exact correct terminology, but think of it as a pipe. But the pipe only opens when there is a certain condition has been met. So this, what you're seeing here, S is the signal which is bound to the outside of that pipe. As a result of it, that pipe has opened up. Now, after opening up, it is allowing the ions to diffuse through it. What sort of ions? Like, for example, there can be calcium ions, there can be potassium, there can be sodium ions, all of these. This is very, very crucial if you've studied a little bit about the way membrane potential is maintained, you probably would be familiar with it. If you've studied ATP generation, remember that you need to have a very proton gradient is very, very crucial in that case. And therefore, in that case, proton pumps work, proton channels, all of these are important. But yeah, each of these terminologies are different. If you're not familiar with it, you need to go and brush a little bit about it, brush up these topics, because what we are aiming to discuss in this class is a little more advanced. So these are very basic molecular biology like definitions. So you just need to do a little bit of effort for that. If after that you don't understand, you can certainly let me know and I'll put together some few slides to discuss it. But yeah, that's the idea. Now the question is, how is it different from a receptor? Now, under most circumstances, a receptor will not allow the direct movement of the ligand into the cell. Instead, what will happen is the ligand will bind to the receptor and it will sort of relay the information inside the cell. So here the first example you see is of a receptor enzyme, which is what we have looked at till now quite extensively, because we have been talking about RTKs or receptor tyrosine kinase. If you had paid attention in the last class, we also discussed um, some other types of enzymes. Are you able to recollect it? We very briefly touched upon a cytosolic class of enzymes. Uh, they are not cell surface receptors. They are intracellular proteins, and they're able to trigger this pathway. Anybody recalls it? Just unmute and let me know.
PIP3,
Sorry.
PIP3 or GPCR.
GPCR is a cell surface receptor. I will be discussing it today. PIP3 was, yes, you're correct. It is, but it's not intracellular. It is a membrane bound. It's just present on the inner leaflet of the plasma membrane. What we have been discussing till now, it's a transmembrane receptor. It's spanning both sides, but it's okay. It's a good attempt. I'll show you the slides maybe then. We did not really get into a lot of detail about it. So if you had looked at this slide, basically, this what you see, STAT enzymes and JAK-STAT pathway. Have you heard about the JAK-STAT pathway? You might not have. If not, it's okay. But the JAK-STAT pathway is actually sorry, just a second. Yeah, the JAK-STAT pathway was the example of an intracellular signaling cascade. And the receptor in that case is also intracellular in nature. It's present inside the cell. It binds to, sometimes it binds to tyrosine kinases as well, and it triggers the entire pathway downstream of it. I just wanted to give you an example, but it's okay if you don't know. So coming back here, yeah. So that was receptor enzyme. What we have discussed till now, they are all present on the cell surface. The third type is for serpentine receptors. This is a new terminology, but we have discussed it and we will be discussing it today. So actually serpentine receptors, they are a bigger family of proteins, but under that one subtype is of GPCRs, which are G protein coupled receptors. And they are also dependent on the binding of an external ligand. And the ligand binding to the receptor helps in activating an intracellular G protein, which regulates an enzyme that generates an intracellular second messenger X. Don't be worried. We'll come to it and we'll talk a little bit about second messengers also today. So That belongs to the superfamily of serpentine receptors. Again, the name serpentine, it's very basic. It basically means it looks like a serpent. And as you can see, because of the nature of GPCRs, where it passes the membrane 7 times, GPCR again is a subfamily of it. But yeah, basically they pass the membrane so many times that they look like snakes. That's why the name serpentine receptor superfamily. Uh, then comes the other sort of subtype where a receptor does not have any intrinsic enzymatic activity. So instead, this is what I was talking about, the JAK-STAT pathway. So these subtypes of receptors, they interact with cytosolic protein kinases. Now, one example of such a kinase is the Janus kinase or JAK kinase. What does that do? That activates a gene regulating protein, it changes gene expression, and then it has an entire cascade that comes into play. So the receptor by itself, look at the diagrams very carefully. They're beautiful diagrams, which will you might superficially just go through, but that is good enough to learn about a lot of stuff. So just look here. If you see this receptor, the cytosolic domain looks different and it is activated. Therefore, it is a receptor enzyme. But here, if you see that the cytosolic domain is also pink in color, that's because it's a simple protein. It's not an enzyme. But instead, what happens when there is binding of this particular blue colored protein, which is itself a kinase, example being JAK kinase, then it triggers a cascade, which then has effect on the cell's mRNA and protein synthesis and so on and so forth. The next class is of adhesion receptors. Now, these adhesion receptors usually they do not have any associated enzymatic activity which is related to them, but they are cell surface proteins and they are receptors in nature. And what do? They bind certain molecules in the ECM or extracellular matrix, which changes the cell conformation and alters its interaction with the cell cytoskeleton. Uh, just to give you a quick recap, do you remember the example? We discussed it in the first class when we were talking about hallmarks. We talked about a certain process where some cell surface adhesion receptors, there's a change in their expression, and that change in expression, the loss or gain of that receptor or the loss of gain of that protein on the cell surface helps in making that process happen. And that process is very crucial and it's considered a hallmark of cancer. Does anybody remember? I want you to keep doing this, otherwise you'll forget what we have discussed in the first class by the end of the course. Anybody, just give it an attempt. I don't think anybody remembers. So yeah, I understand it's been a while, but try to brush up on it. It'll do you good. Um, um yeah, suspense. Do you remember this process, EMT, that we talked about? So here we had talked about all these junction proteins, tight junction, adherence junction, desmosomes, hemidesmosomes. These are all for cell-cell and cell matrix interactions, and they help in keeping the cell intact. Where are the receptor present? Obviously, they're on the cell surface. A receptor, which is necessary to hold two cells together, obviously has to be on the cell surface. It will not do you any good to have that inside, right? So we talked about how there are changes in the expression of these proteins like ecaterins, claudins or cludins, etc., and how that is necessary, that loss of their expression and the gain of some different types of protein expression like encatinin is necessary to help the process known as EMT, which is epithelial to mesenchymal transition. And then the reverse process also takes place, which is MET, that is mesenchymal to epithelial transition. I hope you remember this discussion. It was in the first class. If you didn't attend it, please go back and look at the slides, or you can look at the lecture as well for your understanding. Or if you don't have enough time to do that, just learn about the process online, but learn about it. This is a very, very, very important part of cancer. And if you are planning to work on it, or if you're just interested in it, you want to give exams or something like that in the long run, if you want to be a part of cancer biology in the future, you need to know this. You need to at least know the basics of it, and then eventually you'll learn more. Now, because I have given you some reminder of this, I want somebody to unmute and let me know when exactly this process occurs. Uh, at what stage of cancer progression does it occur? Just try answering whatever you think is correct. Anybody, unmute and go ahead.
Dysplasia.
Sorry.
Dysplasia at
No,
that stage.
no, that's not correct. Anybody else, or you want to try again? Anybody? Anshika, Harsha, or I don't know who this is. Your name is not visible. Just give it a try. Okay. Guys, you need to remember this a little bit. Otherwise, I understand it's a lot of information, but thoda thoda you have to try. So this occurs during metastasis. I had discussed this in the first class. If you're not understanding, you need to let me know. I'll slow down the pace, but you have to retain a little bit. Otherwise, this is not. This is going to be a very pointless journey if you don't remember things. So I'm not saying you need to prepare for it like an exam. I have not planned anything like that right now, um, because it's not really that sort of a course. It's more educational that, rather than it being a certificate course. So that's why it's not to put pressure on you, but if it's better if you remember these things. So basically, it occurs during these sub-steps of metastasis, where from the original tumor that is located at any initial site or the primary site, when the tumor cells they move away or they disseminate, then they have to move into the blood circulation, and then they again have to move out of blood circulation and move into the new tissue. During these steps, EMT to AMT occurs. So EMT is where from the epithelial nature, the cell becomes mesenchymal so that it can move and it can metastasize. And once it has found a new site, what it does is it undergoes the reverse process, that is from the mesenchymal nature, it again becomes epithelial and it forms a new tissue at the new site. So this is what exactly happens. So it's okay if you don't remember things. I understand there are 2 classes in a week, so it's hectic and everybody has their own classes and all. But yeah, it'll be better if you can retain certain things. But it's okay, don't worry. Um, just no. Yeah, so we were here. So we were talking about adhesion receptors. So they bind some extracellular matrix molecules and they're necessary for interaction with cytoskeleton as well. Now, finally, the last subtype, which is what we'll go into a little more detail about, and I actually wanted to give you examples of it, but I think that might be too advanced for students right now. But basically, what it is about is steroid receptors. Now, steroids actually are also different, entire class of molecules by themselves. But when we talk about receptors, particularly, steroid hormones are able to bind receptors. So, why is that necessary? That is because certain hormone, not even certain, most hormone receptors for that matter. Some exceptions are there, like for example, insulin. Insulin is able to bind to insulin receptor, which is a cell surface receptor, and then triggers the entire RTK pathway that we have been discussing till now. But the steroid hormone group of families that includes all of the androgens, all of the estrogens, all those components that we have briefly been talking about, these hormones, they are binding to nuclear receptors. What are these nuclear receptors? They are steroid hormone receptors. And this steroid, why is it able to do so? Because steroid is able to move into the cell upon binding to the receptor, and then it allows the receptor to regulate the expression of some specific genes. How does it do so? We'll come to that. Look at the figure on the right. Before that, let's go through the legend. So steroid hormones and thyroid hormones, retinoids and vitamins, by virtue of their hydrophobicity. Anybody, can you explain what hydrophobicity means? This I'm pretty sure you would know. Give it a try. What is
Mm,
hydrophobicity?
And because the cell membrane is made up of the lipid bilayer,
Hmm.
so uh, the lipid bilayer has like the outside, um, the outside layer. It
Hmm.
is hydrophobic. It has the hydrophilic head, and the inside is the hydrophobic tail. So
Hmm.
because of that, the steroid hormones can enter the cell membrane without any receptor or channel.
Hmm. Correct. So you're absolutely correct. So two components are necessary, the hydrophobicity and its small size, both of those. So a large hydrophobic molecule might find it very difficult to diffuse into the cell, but because steroid hormones are relatively smaller in size, again, relatively being the keyword, they're able to diffuse inside. And like he explained as well, basically because of the nature of the lipid bilayer, as you understand, it is a lipid bilayer. You must have studied the structure of lipids. This we learn in chemistry pretty early on. So you would remember that there is a hydrophobic head and a hydrophilic tails, and they are sorry, hydro. The center of it is the chain, and therefore they are forming this lipid bilayer. So the parts of the lipids that are exposed on the outside, they're in contact with extracellular fluid and the intracellular fluid, whereas the hydrophobic part is conserved within the membrane. And as a result, the interior of the membrane, like if you think about it, like say for example, let me use this as my phone as the example. So these two parts of it are hydrophilic in nature, but the center part of it through which the hormone has to go through, that part is hydrophobic in nature. That's why it is able to go through without any assistance. And because of its small size, it is able to evade any other problems, and therefore steroid hormones diffuse right through, but they won't be able to persist indefinitely. Instead, what happens is once they have diffused through, they bind to the intracellular receptor protein. So some use cytosolic receptors. For example, cortisol does that. Cortisol binds to a cytosolic receptor. The example I was giving of AR, which is androgen receptor, that is also cytosolic in nature. But in some cases, receptors can also be nuclear. So in that case, the hormone has to first diffuse through the plasma membrane, then it has to go through the cytosol, then enter the nucleus, and then within the nucleus, it will bind its receptor. So So these are two subtypes, and the second subtype usually includes the thyroid hormones. So these receptors are structurally related, and they're all considered to be part of the nuclear receptor superfamily. So what happens after that? Here the example given is of cortisol. So cortisol is a hormone. You must have come across it at some point of time. It's it has a lot of different effects, but one of the common reasons why we hear about it a lot nowadays is because it is very significantly linked to stress. People who are very stressed, they have a very high level of cortisol, but you must have studied its basic physiological functions early on. So cortisol is a steroid hormone which is able to diffuse into the cell, binds to its intracellular receptor protein, and the conformational change activates this receptor protein. I hope you understand what conformational change means. Basically, the shape of the receptor and shape of the ligand after binding, it sort of alters a little bit, especially the shape of the receptor alters a little bit, which allows it to now become activated. Now, because of this activation, now what it is able to do, this receptor ligand combination or this ligand bound receptor is now able to move into the nucleus. Now, you might ask, how is it going inside the nucleus? So, the nucleus has certain pores. Again, this is a whole other topic by itself. If you want to learn about it, I also have videos on this. Taught from a separate course very early on. I think I taught it 1.5 or 2 years ago. So there we discussed the basic concepts of cell and molecular biology. So there we have talked about the structure of nucleus, the nuclear membrane, architecture of the nucleus, so lots of other things. But basically, if you are enrolled in some sort of a course which teaches you all of these things, you would learn it at some point of time. So yeah, so the activated receptor and hormone complex or ligand complex can then move inside the nucleus. Now what happens, a very unique and very interesting feature of steroid hormone receptors is that you remember that if this was, say, this was the structure of the receptor before the ligand is bound, then it binds to the receptor. Now the receptor structure changes. What does this do? This change in its conformation or change in its structure helps it to now function as a transcription factor. Now, what does that mean? It means that any protein which is considered to be a transcription factor directly by itself has to have one ability, that is to bind DNA. So there's DNA which you will transcribe, right? So any transcription factor has a DBD or a DNA binding domain. And once this ligand has bound to the receptor, the change in conformation exposes this DNA binding domain. As a result of this, this complex is now able to bind to the DNA, whichever DNA it is supposed to bind. It binds there and then it activates the transcription of the downstream gene. And then it synthesizes RNA, that RNA will get translated and then the job will be done. So this pathway is relatively I'm calling it relatively because there are exceptions to it, but this pathway is relatively less complicated. It has less interruptions, like less checkpoints if we compare it to other pathways. But this is also the reason that these pathways, steroid hormone receptor pathways, are also very, very commonly found to be dysregulated in cancers, very commonly. You'll find a lot of examples. I'll give you the two most immediate examples that we come across every day: breast cancer, prostate cancer, very heavily dependent upon hormonal dysregulation. And therefore, the treatment for these also involves hormonal therapy as well. Other cancers are also, they also carry even ovarian cancer. Basically, organs which are more hormone dependent, steroid hormone dependent, they are more prone to steroid hormone signaling dysregulation, leading to cancer. So, is this understood? Any clarification you need on this? Please let me know. Raise your hand or unmute and go ahead.
No, ma'am. Ma'am, like this nuclear membrane is also double layer membrane. So still the receptor ligand complex passes it without any resistance.
Okay, so this is an entire topic by itself. Uh, like in your childhood, when we were learning biology as kids, if you remember, the nuclear structure is always shown to have these little pores. Do you remember? So they are
Yes,
actually
ma'am.
called nuclear pores. The
Yes,
architecture
okay.
there is very different. So that is the way a cell is formed, that its nucleus is supposed to have pores. Now you might ask why. That is because if you have learned the basic process of transcription, translation, etc., right? Whatever is getting replicated, that is happening within the nucleus. DNA replication is occurring within the nucleus. But once replication has been completed, mRNA has been synthesized. That mRNA has to be brought outside because translation is cytosolic. Translation cannot occur within the nucleus. Therefore, pores are extremely crucial and they're always present in any nucleated cell because that allows the RNA to come out of it. Now, the question that might come is it a free movement? Is it just that anything can diffuse in and diffuse out? No, there a lot of control is put in place. There are certain, like I won't go into too much detail, but basically the think of it like think of it like a filter paper sort of an architecture that is not very specific, but there is still some control that is there. It's not very rigid. It's not like the plasma membrane because the plasma membrane's job is to protect the cell. The nuclear membrane job is to just keep a check on who is coming in and who is coming out, but then it is relatively more relaxed. But even then, there are certain type of proteins which are transporter proteins, two types. I'm not going into details about this, just read about it. There are importins and there are exportins. Importins are proteins which help in importing something into the nucleus. So something from the outside comes into the nucleus. Exportin, something from the nucleus goes outside. So these types of proteins, they help, think of them as luggage bearers. They basically carry the protein with them. They are able to do that by themselves because of the property of that protein itself. It has a certain sequence, all of that. Let's not go there. But basically, these proteins are able to carry whatever luggage or whatever nuclear cargo they have to carry into or out of the cell. So that is their job. So there's a control over there as well. But the steroid hormone receptor, because of its nature, because it has an exposed transcriptional domain, and because of the way it is constructed, that is able to go in unassisted only. I think I'll maybe read about it a little bit more. But yeah, they are able to diffuse by themselves. They're able to go inside by themselves. And once they have moved in, they will do their job. They will transcribe the gene of interest, and more of that protein will be synthesized. Did you understand that?
Yes, ma'am.
You can Google it and you can read, just get an idea. You just see pictures of it. You don't even have to study too much. We can look at it, but then we're already very short of time. But yeah, nuclear membrane architecture is very interesting. It's outstanding. If it's something that's interesting to you, you will love reading about it. So yeah, look at it.
Okay, ma'am.
Okay, so coming back here. Um, so till now we discussed all of these different subtypes. In the next slide, we will be looking at one particular subtype in a little bit more detail, which is about GPCRs. So GPCRs, as the name we already have discussed, are G protein coupled receptors. So what is the big deal about them? GPCR signaling is it's a wonderful area of research, and GPCRs are everywhere in your body, and they're very crucial within the body. This is just a sort of map that is to just give you an insight into how crucial and how diverse their functions are. But you don't have to remember all of this. But basically, there are four types of GPCRs, and they respond to all of these different types of ligands. You might be familiar with the names of some of these, like dopamine, histamine, somatostatin, acetylcholine. All of these are neurotransmitters, serotonin. But these are, some of them are hormones, epinephrine, glucagon. All of these are different types of ligands that bind to GPCRs. Downstream of it, you can see, okay, the color coding is for showing you how much crosstalk there is, but don't worry about it. Again, it's just to give you an idea about how complicated the system within our body is. You don't have to remember everything, but just get an idea about it, that these four types of proteins or types of ligands, G protein mediated signal transduction, involves these downstream pathways like transcription, secretion, cell motility, ion channels and transporters, and metabolic enzymes. All of them are controlled, which helps in controlling all of these different pathways, eventually, which is necessary for normal organismal homeostasis, development, embryonic development and gonadal development as well, and learning and memory. So the entire process of this G protein mediated signal transduction obviously is very crucial. That's the point I wanted to convey. Now let's get into details about the pathway. So there are three types of components of any G protein signal transduction pathway. The first, obviously, is a receptor. The second is G protein, and the third is effector. Let me just go to the next slide. Yeah. So you will get more detail about this in this slide, but let's first learn the definitions of the two. I'm just giving you this as a reference. So to come back here, what are these components? The first one, like I said, is the receptor. This subclass of receptor involves integral membrane proteins with 7 transmembrane segments. Now you might say, what is integral? Integral means something which is only a part of the membrane. Look here. Okay, this representation might not be very clear. Maybe we'll go to this representation. Yeah. So basically, the entire protein is only present within the plasma membrane. It's not a transmembrane protein. There's a difference between transmembrane, intracellular, extracellular, and integral. So integral is only part of the membrane. So GPCRs are integral membrane proteins, and they are 7 pass or 7 transmembrane segments are present with them, and they also therefore belong to the serpentine family of transcription, serpentine family of receptors. They have binding sites for very diverse ligands like hormones, odorants, tastants, light. More than 907 human GPCRs. Now, keep in mind, this is a very old data. This is from 2002. But yeah, it's a published paper. So that's what I took. You can go into very recent data. There are thousands of GPCRs which have been identified now. And genome sequencing has revealed a very vast number of new family members, which are excellent targets for drugs to be discovered. Again, this is something you should understand, that because this pathway is so crucial, the more crucial a pathway, the better it can look as a target. So if you think of, when I say target, it means for a cancer cell, right? So if a particular, not even cancer, for any disease for that matter, but yeah, mostly we look at signaling pathways for cancer itself. So more crucial a pathway, the more promising it can look to you as a target. But at the same time, what you need to understand is you also need to keep in mind that if a particular pathway is very crucial for a cell to survive, the specificity with which you have to shut off that pathway has to be very high. Say, for example, if a particular GPCR pathway is necessary for a cell to divide, for a normal cell to divide. Without it, pathway will be completely shut off. That is the major approach that is taken for any cancer treatment as well. Now, if you are trying to stop cell division, then you have to keep in mind that this targeting or this drug, whatever drug you're using, is only going to affect a cancer cell and not the division of a normal cell, because normal cell division is necessary. Now, this is the major problem that we face in cancer, that this specificity is lost. And therefore, you see that people have so many different types of these off-target effects. And most commonly, what you see for chemotherapy patients, they often suffer from these problems that their stem cells are lost, or people who have been subject to radiation as part of cancer treatment, that they lose stem cells. And stem cells are very crucial for hair. So hair is the first thing you see people lose as a side effect of treatment, because that is necessary. It needs to be constantly dividing, and uh, that is one of the first things that get affected. Then nails start getting weaker, eyelashes are lost. So basically these are the most common things that you see happen in people. So therefore, targeting a pathway in a very specific manner is a challenge that we are facing till date. And what has happened over time and what is the current area of research is something we will be discussing in the next 2 or 3 classes, maximum 3 classes. We'll try to wrap it up by then. But yeah, that's the plan for the upcoming classes. Now, coming back to this slide, apart from receptors, the two other components are G proteins. So G proteins, like you saw, they are of all of these different types. G is, I think, for globular only, so globular protein family or G protein family. So they are trimeric complexes. Trimeric means it has 3 subcomponents, which are alpha, beta, gamma. And this is actually a GTPase switch protein. What does this mean? It binds to GTP, and it acts as a GTPase, and it cleaves GTP to generate GDP. And no, sorry, that's a Jeff. Sorry, I'm very sorry. It binds to GTP and it is able to exchange or it is able to bind to GDP and GTP both. I'll show you that. I might have confused you with this now, but you'll see and you'll understand that. And it can often be attached to membrane as well. Apart from that, effectors, which are downstream of the signaling pathway, include adenylate cyclase, phospholipase C, phosphodiesterase, ion channels, and effectors have control levels of secondary messengers like cyclic AMP, cyclic GMP, DAG, and IP3. We'll come to all of it. What's the time? Just check. Okay, we have time on hand. So basically, what exactly happens downstream of this signaling pathway? Yeah. So look at this particular pathway. Basically, the example given here is of epinephrine. Epinephrine binds to its specific receptor, which is a GPCR. See how it is arranged. So you have 1, 2, 3, 4, 5, 6, and 7. 7 times it has crossed the membrane. And after that, the entire protein is limited only within the membrane. Therefore, it is an integral membrane protein. What you see here is a sort of a cavity to which epinephrine can bind, and therefore the ligand is binding to its specific receptor. Following this, what you see attached or linked very closely to it, sorry. Yeah, so what you find very closely associated or coupled with the receptor is a G protein. Hence the name G protein coupled receptor. So this is the GPCR, but this is the G protein. Now here you see GS written. So GS is indicative of the first subtype, like you saw here, the GS subtype, the G stimulatory subtype. So that is present close to this epinephrine receptor. It has three components, G alpha, G beta, and G gamma. Now, beta gamma are present together as one subunit and alpha is present separately. So what happens here? Uh, the occupied receptor, which means that once the ligand has bound to the receptor, step one is that ligand binds to its receptor. Step two, the occupied receptor causes replacement of the GDP bound to GS by GDP. So GDP bound, GDP is, I hope you're aware of it, it has a diphosphate, guanyl diphosphate, whereas GTP is a triphosphate. So GTP is the activated version, GDP is the switched off version of the same protein. So once the hormone or once epinephrine has bound to the receptor, then the alpha, beta, gamma subunits of the G protein, which is coupled to that receptor, will dissociate. How will it dissociate? Alpha subunit moves away, whereas beta, gamma subunits stay together. Now, this GS alpha subunit, which has exchanged or has switched its GTP, will then move and it will, like, it has gained GTP and it has removed GTP. Then what will happen? It will become activated. The GTP bound form of the G protein is active. And what does it do? It activates the associated effector protein, which was adenylyl cyclase, shown here as AC. What does that do? Adenylyl cyclase catalyzes the formation of cyclic AMP. What is this? So what this does, adenylyl cyclase. Cyclase means it does something cyclic in manner, or it is converting something into a cyclic compound. How is it doing so? It takes ATP, which you know is the energy currency of the cell, and it converts it into cyclic AMP. This is the job of the enzyme. Adenylyl cyclase is the enzyme which does this job. Cyclic AMP will then act as a second messenger. This is something somebody had asked in the last class. I think Harsha or Ankit, one of them had asked. So basically, cyclic AMP is formed, and then this cyclic AMP is able to execute all the downstream jobs. What kind of jobs can it do? It can activate protein kinase A, it can get degraded, it can form 5' AMP, or it can again get circularized and form ATP again. So a lot of things can happen. Another pathway that is often activated is via phospholipase C. I think I had, yeah. Maybe we'll come to this later. Let's just clarify this first. Again, it'll be too much information otherwise. So what exactly happens in this pathway? Just easy to remember. It'll just think of it like a flowchart. First, you have a ligand. Here the example given is epinephrine. Epinephrine binds to its receptor. What sort of receptor? GPCR. What does that mean? It is a receptor that is coupled to a G protein. Where is that G protein? It is closely present, very closely associated with the receptor protein, but it is not like, it's not a bond, but it's just in close association with that protein. Where in the membrane is it present? It is present in the inner leaflet. If you look very closely, that's what the diagram also shows. So you have this lipid bilayer, and within that, in the inner leaflet of the lipid bilayer, the G protein is present. Yeah. So what happens after epinephrine binds? The binding of epinephrine causes the GDP bound inactive form of G alpha to dissociate from beta gamma. So beta gamma stays behind, but alpha subunit moves ahead, binds to adenylyl cyclase and activates it. Adenylyl cyclase, again, ASE is coming in, which means it is an enzyme. What sort of enzyme is it? It takes ATP and converts it into cyclic AMP. What does that mean? It removes two phosphates and it cyclizes the entire molecule, and this cyclic AMP can then execute the downstream effects. Keep in mind that cyclic AMP is a second messenger as well. So this also indicates similar things only, just a easier representation of the same thing. So the activated beta gamma protein stays behind, but the alpha subunit of the G protein, which is GTP bound, is able to move ahead and it is able to interact with the activated enzyme. Example being adenylyl cyclase. So this adenylyl cyclase then converts ATP into cyclic AMP. What does that do? Many intracellular messenger molecules are then able to diffuse very widely to act on target proteins in various parts of the cell. The two most common enzymes that are activated by G proteins include adenylyl cyclase, which does this job like we discussed, and secondly, phospholipase C. This cleaves a lipid, example being inositol phospholipid, into IP3, which you are familiar with by now, and also DAG or diacylglycerol, a lipid in the membrane. Both IP3 and DAG, they are second messengers. So cyclic AMP, IP3 and DAG, all three of them are second messengers, and what their job is to diffuse into the cell, bind or act on target proteins present in various parts of the cell and execute their job. Any doubts you have from this diagram, let me know. We'll discuss it again. Just unmute and let me know before we move ahead. This, or in fact, what we have discussed till now, any queries, anything you are uncertain about, just unmute and let me know.
Ma'am, can you please explain the seventh step, that is the CAMP is degraded and reversing the activation of PKA?
Oh, that is a separate pathway. So till now, what we have discussed till now is the fact that cyclic AMP is produced. There are multiple things that can happen to cyclic AMP. One is that it is forming 5' AMP, and that 5' AMP will again go back. That means if you have studied, you might have studied about the two structures of glucose, right? For example, there is one structure which is cyclic in nature, and there is one which is an open chain structure. So similar to that, cyclic AMP means basically it is an AMP molecule which has become a complete, which has closed the ring on itself. So that can then be removed, or that can be linearized by cyclic nucleotide phosphodiesterase, which cleaves that backbone and it linearizes it. So this is one possible pathway. So alternatively, what can happen is that if there is the default pathway, if there is a lot of cyclic AMP, what will it do? It will activate protein kinase A. Protein kinase A, the kinase, it's a kinase by nature. It is responsible for activating or activating certain pathways by phosphorylating cell proteins. And these proteins, which will be phosphorylated, that depends upon which hormone has bound here. In this case, it is epinephrine, or which ligand has bound here. So that is its default job. For cyclic AMP to be produced, cyclic AMP to activate protein kinase A to go ahead and phosphorylate the downstream proteins. Now, when there is cyclic nucleotide phosphodiesterase, which is present, what will it do? It will start cleaving cyclic AMP, and it will start generating 5' linearized AMP, normal AMP. Then this AMP will again be utilized by certain other enzymes, some kinases, which can generate from AMP, it can generate ATP, or basically ATP synthesis will occur eventually in very energy intensive steps, and then the cycle will be completed again. So basically this sort of serves as a check or a control over how much protein kinase activation is allowed by degradation of cyclic AMP when necessary. I think there's a feedback loop that was also there. I cannot recollect it at the moment. I'll go and look at it. But it's a negative control over the usage of cyclic AMP to restore the balance of cyclic AMP and ATP within the cell. Okay. Did you understand that?
Yes, ma'am. Yes.
Anybody else? Any doubts? Nobody else seems to be interacting today. Anything you want to share or anything you want clarified?
Ma'am, there are no doubts, but it's just that it's a little overwhelming. So
Okay.
it need a little time to actually absorb it all.
Okay, I understand. See, that's the thing. So the idea of the course was to go for more advanced concepts, which is what I had mentioned, but I understand that everybody has different backgrounds, so maybe you're not all on the same page. Just let me know, did you understand the first class where I talked about the hallmarks of cancer? Was that okay with everybody?
Yes, ma'am.
Yes, ma'am.
Okay, so I think
Yes, ma'am.
signaling is a little more challenging for everyone. Okay, no problem. Don't see, first of all, keep in mind you are not under any pressure. You're just learning this for yourself and for your future, whatever you want to do. So that's why I went back on this topic, because it's a little more complicated. And what I had planned initially as part of the course structure, it's not in this domain exactly. It's not going to be very related to cell signaling, but it'll be more to keep you abreast of what is happening in cancer around the world. So don't worry about it. This you understand. Even if you don't understand it today, you can come back. You can always discuss it with me. I keep my mail open for any queries, anything you want to learn. I completely understand this is overwhelming. I have learned cell signaling over a lot of weeks, over a lot of classes. It will take you that much time as well, especially if you're not familiar with it. But it's okay. Just understand this for your own sake, for your just for your knowledge. Keep it to that. And any point which is not clear, let me know. I'm not going to go into more details about pathways. What we have learned till now are the basic subtypes about different pathways. The first one, the first example was of receptor tyrosine kinase pathways, right? They are tyrosine containing receptors, as the name suggests, and they are cell surface receptors based on which there is a downstream cascade which is triggered. That is the first subtype we learned. The second subtype that I have mentioned very briefly is about steroid hormone receptors, right? So steroid hormones depend upon the binding of a ligand, and therefore downstream of it, transcription factor activity comes in, and then they're able to execute their job. Third type of pathway, which I'm discussing now, is of GPCR, which are G protein coupled receptors, and therefore they are dependent upon the presence of a G protein, and they also function in a certain manner. So these are the three types of pathway. We'll keep it to that. Okay, so don't worry. I had planned on going into more specifics about pathways, but it is unnecessary, and it is a lot of molecular biology that you need to know. So don't get worried. Uh, Let me know if you want it to be a little more simplified. I had considered getting you videos, but I think it's a waste of time. Instead, maybe I can drop some YouTube links, because there are wonderful animations that you'll find online to understand how all these pathways work. But conventionally, people have learned from schematics, so that's what I've kept. But yeah, again, I just want to reiterate, don't worry, don't take too much stress about this. If you eventually work on cancer, you will learn it one way or the other. And knowledge is endless. Cancer is endless. So there's no upper limit to learning. So it's okay. Just take it as something you want to know. And what I said about keeping in mind, the hallmark part is because hallmarks are something which is very relevant to what I wanted to discuss in this course. Cancer cell signaling can be a course by itself. So here we are just looking at it very uber se. But it's okay. Don't worry. If you have more doubts, you let me know. I'll share more material for you. But if it is something which is too overwhelming for you, keep it at this. Okay, just learn this much for now, and you can think about it later. If you find it interesting, you can learn about it in the future. Okay, so don't worry. And if you have any queries, please drop me an email. I can talk to you personally as well. Don't hesitate. Okay, so this is about GPCRs. This just shows what I have just said in more detailed schematics. So basically, it is you have your signal molecule, epinephrine, binding to the receptor, and then the receptor is leading to the activation of the coupled G protein. The activated subunit of the G protein goes and binds to the activated adenylyl cyclase. What does that do? That converts ATP into cyclic AMP. Cyclic AMP, what does that do? It goes and activates protein kinase A from inactive to active state. What does that do? That goes inside the nucleus. Oh, see here it's mentioned nuclear pore. That's what we were talking about before. So this is a sort of a channel within the nucleus that is present. Protein kinase A goes and activates. This is just an example. It's not as simple as you can see here. A lot of things happen inside as well, but basically this is the outcome. So protein kinase A upon activation binds to some gene regulatory protein that is getting activated, that is influencing the transcription of some gene, which is leading to more and more production of that RNA. So this is the overall outline. I'm going to repeat it one more time. First, hormone binds, signal molecule binds, to the receptor present on cell surface. Follow the diagram. You'll remember it eventually. We were taught this exactly the same way. The same thing was repeated 20 times until it was like, etched in our brain. So the signal molecule binds to the receptor. Receptor activated, as a result of which the G protein, which is associated here, it has not been shown, but you understand it by now. The G protein, which is present along with it, the beta gamma subunits stay behind, the alpha subunit moves ahead. The alpha subunit goes and activates adenylyl cyclase, which is present on the membrane. Sorry, adenylyl cyclase is responsible. The name itself is clear. Adenylyl cyclase, adenylate residue is getting cyclized. What does it take as the substrate? ATP converts it into cyclic AMP because it's a cyclase. Cyclic AMP is a second messenger. What does it do? One of its major downstream actions is that it binds to or it converts or it activates inactive protein kinase A and it activates it. Activated protein kinase A. The name is it will work on some protein. Kinase means it will put some phosphate group. What will protein kinase A do? It will go inside the nucleus. It will bind to some protein, gene regulatory protein, and it will activate it. This protein will then influence downstream transcription. Okay? So there are 6-7 steps which are happening. Step 1, binding. Step 2, G protein dissociation. Step 3, activation of adenylate cyclase. Step 4, conversion of ATP into cyclic AMP. Step 5, activation of protein kinase A. Step 6, activated protein kinase A binding to gene regulatory protein. Step 7, increased transcription. 7 steps. Okay? Not as complicated. Think about it. The more it's your first time learning about something, it will of course seem like, oh my God, so much information. But once you learn, once you have written it down, it'll be perfectly fine. Don't. The other way in which second messengers work, I think somebody had asked it last year, I've forgotten who, but not another way, but just another representation to show you how second messengers work. So basically you have the hormone binding to the receptor, the associated G protein from GTP, it is like GTP is coming in, GTP is going out. As a result of this, the activated receptor binds the other possible pathway that I talked about to phospholipase C. The first one we talked about was to adenylyl cyclase. The other possible pathway was phospholipase C. What does the name mean? Phospholipase C. It is cleaving some sort of a phospholipid unit. What unit is that? PIP2. What does it do to that? It releases IP3 from it or inositol triphosphate from it. And alternatively, what you also see, it generates two types of compounds, PIP2. It generates DAG, which is diacylglycerol. Again, don't stress about it. Just look at it like somebody is just telling you something interesting or something overwhelming, whatever suits you. So basically, it is generating DAG, which is diacylglycerol, and simultaneously also releasing calcium. DAG and IP3. Sorry, I messed that up. Phospholipase C is cleaving PIP2 and generating DAG as well as IP3. IP3 is then stimulating the release of calcium from the endoplasmic reticulum. So there's just a section shown, but the entire unit is actually the ER, the endoplasmic reticulum. From the lumen of the endoplasmic reticulum, lumen means the interior side of the endoplasmic reticulum, calcium is able to move out. And this is a major change that occurs downstream of cell signaling. Like somebody had asked, right, what exactly happens after, why are we calling it a second messenger? What happens after it is present? So calcium signaling within any cell is a very finely regulated and a very crucial process. Why does it happen? That's because the endoplasmic reticulum or the ER is a storehouse of calcium. So under normal circumstances, very high concentration of calcium is maintained within the ER. When there is this sort of a stimulus that some sort of a change has happened as a result of which IP3 has been released, then IP3 commands the ER or through some sort of channels. For example, this is a calcium channel. Again, this is where you see what is the difference between a receptor and a channel. You can see that there is a port through which the ion can diffuse out. So IP3 stimulates the release of calcium, and once this calcium has been released, it it executes its downstream effects. One of which, for example, is that it binds to some sort of a kinase, which is a calcium calmodulin complex. Don't have to remember it. But basically, that triggers the phosphorylation of some proteins, or sometimes calcium can enter in via DAG, calcium can enter into the cell, and via IP3 calcium can exit from the ER into the cytosol. So basically what is happening is that there is a shift in the calcium concentration inside the cell, and that is able to execute some downstream effects, one of which is the phosphorylation of some target proteins, and whatever is the job downstream of it, that happens. So overall, if we sum it up, we started off with the same process for GPCR. I'm not going to repeat that. I've said it too many times now. But basically, activation of GPCR can have downstream effect on two types of enzymes, either adenylyl cyclase. The name the name is explanatory, self-explanatory. It takes some adenylyl molecule and it cyclizes it, which means ATP to cyclic AMP. The other one is phospholipase C. It cleaves, or it takes a phospholipid and cuts it, and it generates DAG and IP3. Both of them eventually help in the release of calcium to increase the cytosolic calcium concentration. Why does it do so? Calcium is necessary to control the regulation of some pathways, one of which is the phosphorylation of some target proteins or target enzymes, which will go and complete the stimulation of that pathway. This is the overall idea. Did you understand it? Some part of it did you understand, even if the whole of it was very, you don't have to remember right now, you just need to understand what I'm saying. Was it somewhat okay? Did you understand some parts?
Yes, ma'am. Understood.
Anshika, did you?
Yes, ma'am. Yes, ma'am.
Okay, cool. Okay. So I'll not go into this right now. It looks crazy, I guess. But let me just move ahead. Hmm. Okay, I think I'm going to keep targeting for the next class. There was a lot of information I gave today, and it was very quick as well. So let's go back to this. Yes, let's go back. Let's revise today's class. What did we see? Key concepts. We learned the different properties of any cell signaling pathway. We learned about the different types of receptors, intracellular cell surface receptors. We understood their characteristic features. We understood steroid hormone pathways. We understood G protein signal transduction, and we looked at the types, the two subtypes of G protein signal transduction as well. Right? That is all we have discussed. It was not a lot, it was a little bit, but yeah, it was a lot of new concepts. What I had also thought of discussing with you, but I'll not go into details of it because it's okay. You don't need to know the signaling pathway. But basically, I want you to learn about what TB53 does. Okay, let me just stop sharing. Let's just have a discussion. If you have learned and if you have attended the previous lectures, you would remember we have talked about oncogenes and tumor suppressors, right? So there are some proteins which are supposed to protect the cell and defend the cell under normal circumstances. So those subgroup of proteins usually belong to the category which we know to be as tumor suppressor proteins within the cell. Why is that necessary? Tumor suppressor is the name that we have given over time. Their discovery has been made in such a way that they have been found to be absent in cancer cells, or absent or modified or reduced, or some sort of change has occurred in the tumor suppressor. One major example that happens, I think I'll show you that slide because it will give you an idea of how crazy it is. Just give me 1 second. Yep, this one. So just look at this slide. Basically, TP53 is very commonly known to be guardian of the genome. Why is it called so? Because of its job. Let's just put it simply like that. It has so many functions. Under normal circumstances, it is supposed to take care of the genome. It plays a very crucial role in the cell division process during cell cycle, whereby it basically keeps a check on the fact that if the during the process of cell division, during the process of DNA replication, if there has been anything, any change, any mistake within the process, and the genome is now modified, then TP53 under normal circumstances will not allow such a cell to proceed further. So for example, if there is cell A, a mother cell, a parent cell, which is dividing, and that cell has, face some sort of an external stimulus. Either there has been some ionizing radiation, some X-ray exposure, some mutagen, or for whatever reason, their genome has been affected. And it is carrying some very crucial mutations. Then, when that cell divides, at the time of cell division, during certain sub-steps, I think I have that. Yeah. So with this schematic, you can just look at it. You don't have to worry too much about it. But basically, whenever there is some sort of a problem within the genome, that is what is represented here as DNA damage. But apart from that, what also is also found to be relevant in context of P53 is hypoxia. Hypoxia means very lack of oxygen. If there is an oncogene, if there is oxidative stress means a lot of oxygen radicals are produced, reactive oxygen species or oxygen radicals are produced inside the cell or any other cell or any other stress when that is present. Then under that circumstances, P53 comes in and it has some major jobs to play. But how do I explain? It is stabilized and its job is to regulate a number of different pathways. And one of its major jobs, because it is also a transcription factor, is to control the expression of some downstream signaling pathways. This is its normal job. Think of it in this way. So whenever there is a cell stress, whenever there is DNA damage, whenever there is all of these other processes that are, just a hectic situation for the cell, then TP53 comes in. And what I called it to be a guardian of the genome is because it has these, it protects against DNA damage of all different types. So then what it does is it controls, it regulates, it ensures the cell is healthy, and only then it allows the cell to proceed further for division. Even when a cell is not dividing, even under normal circumstances, TP53 is necessary, and it's a master regulator of many pathways, and it helps in maintaining cell health or normal cell behavior, cell state. Now, what happens when there are mutations in TP53? Then this problem is completely lost. So basically, TP53 levels in a normal cell is kept under a lot of check. So you understand, as good as P53 may seem to you that, oh, it is helping in, it is curing everything, it is removing all problems. But then unlimited production of P53 can also be very dangerous. And it can be a huge advantage for a cancer cell. So irrespective of what it carries, if a particular protein is just, helping it survive under all circumstances, then that will be a problem. So in a normal cell, the levels of P53 are very strongly kept under check. It is done by these proteins, these MDM2 and MDMX. Don't need to remember too much about it, but P53 levels are basically controlled. When this control is lost, then it is a huge problem. Now the question is, why will there be a loss in its control? That is because P53 is found to be very, very commonly mutated. Like if you look at any cohort, any population, you will find that at least 50%, if not more, patients carry a mutation in P53. It is definitely there, almost definitely there. Um, I'm not saying in every individual, but in a large proportion of individuals, P53 is mutated. And like you can see, this statistic here shows exactly that. Mutations can be of different type. The most common one is missense mutation. I hope you know about these. Can somebody just unmute and let me know if you know the difference between different types of mutations?
Yes, ma'am. No.
Okay, anyone?
Yes, ma'am.
Okay, good. So missense, frameshift, nonsense, all of these different types of mutations can be present. And you can see the percentage, the approximate percent. This is from one study, of course, but you can see the approximate percentage in which you can find the mutation to be present. And what does mutant TP53 do? Basically, it has a huge effect on transcriptional activity, on protein-protein interactions, and also what's this? The secretome, mutant p53 secretome. Basically, it has a lot of different types of effects. Like, for example, loss of wild type function is completely gone. So normal cell function, normal protection of the cell is gone. Unfolded proteins, which means cells that have improper or wrong protein synthesized, even they will go ahead and they will keep on dividing. Genome stability is lost. Genome becomes unstable, which means that even if the genome is broken somewhere, carries random mutations, such a cell will keep on dividing. And the more number of times it divides, the more number of mutations it carries. And therefore, that is how it helps progression of cancer. Then you have metabolic reprogramming, which means it leads to change in the programming of the cell, as a result of which it becomes cancerous. Tumor microenvironment can be affected. Tumor formation can occur. Invasion metastasis occurs. Drug resistance can be held. Proteasomal activity is lost. Dominant negative effects are lost. So basically, it has some gain of function effects. Gain of function means that it helps in acquisition of a cancerous state, and some loss of function events are occurring, which are also helping in the same. Basically, both types of effects are eventually culminating into a cancerous state. So that is how the TP53 signaling pathway affects it. Here I had just looked at a little bit detail about targeting pathways, but we'll take it up in the next class, don't worry. But yeah, this is a question I want you to read about a little bit. The three questions. One, TP53 signaling pathways, do they have any pro-tumorigenic role in any cancer? So why have I asked this is because you can see that it's not supposed to help tumor formation, right? So TP53 signaling pathway is supposed to keep a check. But there are some reports about certain cancers where P53 leads to the normal functioning of TP53 leads to acquisition of cancer. Just look at it, just Google it. You'll find some answer to it. So what are the off-target or non-specific side effects of targeting oncogenic signaling pathways? Well, I have discussed that already in a lot of detail. If you were to improve the specificity and efficacy of existing drugs, how would you do so? So we'll address this question after we talk about therapeutic targeting of signaling pathways and examples of some drugs. So we'll do that in the next class. Anybody who was not present in the beginning, let me reiterate. I'll also drop an email. We'll have the next class on Tuesday. I think that is 4th March. Yeah, we'll have that on 4th March. We'll start from our regular time, 5 p.m. In any case, if there's any problem, it shouldn't be there. But in case there's any problem and we need to postpone the class a little bit, I will let you know. I'll let you know a little bit in advance. Don't worry. So, and we'll cover these topics. We'll just spend a few more minutes, 15-20 minutes extra, and we'll just discuss whatever is left. And we'll wrap up signaling hopefully in the next class. And the class after, we'll take up the next idea, which is about cancer diagnostics. So that will be a little more relaxed. It will not be this signaling intensive. It will not be that memory intensive class. So that is what we're going to end on today. Any questions, any doubts, any suggestions, just let me know. Anything you want to share? No? Okay, I'll stop recording. Just